Great, thanks very much for the invitation to participate. Um, this is a talk I've never given before. All the slides were newly created, so there's definitely going to be mistakes, and apologies for that. But the goal is to basically describe kind of the nuts and bolts of how one sets up a large-scale multi-center collaboration with an eye to identifying, common pitfalls that can occur along the way. It largely tracks my experience with establishing a couple consortia related to some quantitative traits. But I would say if I harken back to my postdoc days when I was trying to decide what projects to pursue in the complex trait genetic space related to cardiovascular diseases, one of the things that I considered at the time was, did I want to study dichotomous diseases? These are things that patients experience adversely, and they become cases, and one needs case control collections to be able to study them. My interests were in dilated cardiomyopathy related to my clinical interest in sudden cardiac death. But at the time that I was planning studies during my postdoc, which was from 2002 to about 2007, but most of this was set up by like 2005. There were very small numbers of collections of cases and controls that had been published in the literature. And so I was pretty concerned about having adequate power. And so, like many, I explored quantitative traits, and specifically, quantitative traits have an advantage that every single person in a study has one. They can be modeled if they're normally distributed quite easily. Many are not, but in fact, that's something that's easy to ascertain. Continuous blood pressure on a millimeter of mercury scale is a good example. If one wanted to study hypertension, that would be a dichotomization of this trait, and the continuous blood pressure measure is the way in which that trait is identified, or that condition is identified. Another would be, for example, QT interval measurable on the electrocardiogram on the millisecond scale. Every single person in the study has one, so you can collect and ascertain many people. And I'm using these ballpark numbers of on the order of thousands to tens of thousands based on the mid-aughts. Of course, now with very large-scale biobanks, we're now talking about hundreds of thousands, but I think the considerations remain the same. And before getting started, you really need to consider what's known about the genetic architecture of these conditions. Are they Mendelian syndromes that are thought to be mostly caused by rare, mostly coding variation, in which one would need to do large-scale sequencing, or is it a complex trait that is likely to aggregate effects from hundreds of common variants, as well as rare variants, as well as environmental factors, in which case genotyping arrays can be much more efficient ways to have a large enough study to be adequately powered. And I think it's really important to be pretty formal about it. I think in the modern era, people are pretty familiar with very large scale data sets, but I can tell you that in the early 2000s, some of the most high impact papers to come out of the Broad were papers that are the precursor to the Broad, were papers that essentially debunked candidate gene, candidate variants literature that was just cluttered with false positive associations due to wildly inappropriate significance testing. And when large sample sizes are needed, one really needs to determine how you're going to be able to assemble those. If it's a niche condition, or a rare condition, or a niche phenotype that has to be very precisely measured, it's possible that new recruitment would be needed, which has a very long time scale. And today I'll be talking mostly about existing phenotype collection and genotype collections. And the focus of the talk will essentially be, how do you scale up for large scale collaborations which are in many cases needed to be able to detect effects that might be quite modest. So essentially, I think in broad strokes, one needs to define the goals for what one wants to accomplish, establish a set of rules for participation. I'll talk about some of the operational considerations, including the conduct of the study and timelines, as well as implications for manuscripts or grants. But again, this is going to be very nuts and bolts, very focused on logistics and tactics, more so than overall scientific strategy. I think it's very useful at the beginning to develop some sort of mission statement. Typically, this is developed by soliciting input from a handful of colleagues who are, familiar with the phenotype of interest. Some sort of one-pager in which there's a brief description of the goals of consortium and what the initial project focus would be. This is helpful to shop around to potential participants who might contribute to the study. Identification of cohorts who might be approached is readily done by comprehensive literature searches and then word of mouth, and circulation of this this one-pager preliminary description essentially can be done to identify representatives of different cohorts or identify the appropriate member of a cohort who might be interested to participate. And then once a large enough group has been assembled, even if it's smaller than the final group that will contribute, then arranging some sort of planning teleconference to gather a consensus on the goals of the project. And so I think it's important at the outset to really establish rules of the road for how the consortium will function. And this is mostly guided by mistakes that have been made along the way by myself and others. And you kind of tend to learn what the typical hiccups are. So number one, one needs to delegate to a relatively small group of people to draft some sort of detailed memorandum of understanding, which commonly can be modeled on prior consortia, and which specifically addresses a range of issues that are relevant to cohorts deciding whether they want to participate in the study or not. And many of these issues have been worked out by large kind of consortial efforts, but I think the focus of this will not be so much how do these large, well-established consortia continue to function, but rather how does one set them up. And so it's important to remember that when conducting human subjects research, even if it doesn't technically meet the definitions of human subject research, depending on the analysis, confidentiality is really important, but to protect the interests of the participating studies and the member study subjects who have participated in their studies. Transparency is really important. Most consortia are comprised of many of the key people in the field who have been historically contributing to these conditions or the study of these traits or diseases, and so in some way they're all competitors in the sense of trying to make discoveries in a timely manner and not be scooped. And so it's important to recognize that to get all those people to work together, it's really important to establish general principles of transparency, and that usually means clearly articulating when one's own institution-specific group may be working on a project that has some overlap with the overall consortium project. It's important to kind of declare those in at least broad form, especially if there are papers that are in process, so that folks know what's coming. It's important to establish principles of authorship, and this, it's not so easy to. There's a tension between allocating authorship or identifying who the leads will be to do the heavy lifting on a project, and figure out exactly what the order of authors will be. So general principles at the outset are quite important to articulate, and then can be drawn on as questions of authorship are mediated down the line. There needs to be some mechanism for conflict resolution, which inevitably arises in various ways, and I'll touch on that subsequently. It's important to establish timelines that have a reasonable likelihood of being achieved, so that folks, when they sign on to a project, have a sense of what the lifespan of the project is likely to be, because obviously the participants and individual investigators may have their own constraints of the amount of time they can allocate to training, and or the amount of time they'll be at a given institution. It's important to identify who the responsible parties are. Typically, a cohort or a site PI is identified who kind of signs off on the agreement to participate in the consortium, and then typically will supervise or work with an analyst who often is a trainee, but in fact at many sites the PI and the analyst are one and the same. So there needs to be flexibility for how those things are structured at the local site level. And then lastly, clarification about what the acceptable uses of the data are and what sorts of protection there are for the consortium who's developing the data. And I think folks can appreciate, with active, very large scale projects, many of which have taken place at the Broad, for example, there's periodic public updates of the underlying data that are done in advance of preparation of a manuscript. And I think one can appreciate the tension between releasing as much in as timely a manner to allow scientific investigation to proceed expeditiously, but also the potential threat to recognition of the contribution of the people generating the data. So laying out the acceptable uses of data, whether it's usable prior to publication or only after publication, and what sorts of protection the consortium have. And if you think about it from the cohort perspective, they have to contribute a certain amount of effort. The central analysis group has to contribute a certain amount of effort, and if that effort is essentially preempted by premature, for example, publication of a high impact paper that takes advantage of the data, then there's a threat to the recognition of that effort in the form of a paper or for grants. And I am open to answering questions at any point along the way. Again, this is just kind of mostly guided by experience and different problems that have arisen, but there may be different models that folks are familiar with or think recommend themselves. And you can use the chat for that, or the Q&A, which I don't have the Q&A. Now I have the Q&A up. So I'll keep track of those, but feel free to jump in and ask questions. So often, depending on how large the consortium is, having a steering committee can be very helpful. The plan is essentially that a steering committee provides overall leadership or overall guidance of the project, but not specifically all the granular logistical details. It's important that the steering committee be not so big that it's unable to achieve a consensus, or even preventing the ability to schedule meetings. So I've been in some consortia where we might have 80 different analysis sites spread across multiple continents, and there are time zone issues. If you set up a steering committee so that the site PI for each location is a member of the steering committee, then all of a sudden you're talking about 80 people on a steering committee, and it's all those different people to chase down, and it can be very unwieldy. So I would say something on the order of 5 to 10 for a new consortium, and maybe something like 10 to 20 for a more established one that's been around for a while, are reasonable ballparks. It can be a forum to mediate conflicts when they arise, which inevitably happens. It's a place to inform the overall leadership. It's generally comprised of more senior, experienced people who have experience across multiple long-term projects and are able to avoid some of the kind of rookie mistakes that can be made and provide general guidance. It can allow consolidation of decision-making. It's important to make the composition of the steering committee equitable and to have representation of various cohorts and different interests. You know, I've been in some consortia where, you know, there might be phenotype or subject matter experts or disease experts who try to run the steering committee, but then none of the people who are have very detailed understanding of analysis are a part. And you know, you really need to span the spectrum of expertises in your steering committee to provide effective steerage of a project. And the steering committee can have, you know, usually have chairs, co-chairs, and these folks can provide, for example, letters of support for grants or training grants or other things to kind of represent the project. And then one should have working groups, and these essentially report up to the steering committee. Examples of working groups might be analysis teams. You know, there are multiple levels of organization here. One is at the cohort level. So if it's a study that has, say, 20 different contributing cohorts, then there will be analyses that are happening in a distributed and parallel manner at each of those sites. And then there will typically be coordinating centers that receive data and are performing central analytic functions. And I'll try to convince you that ideally at least two sites or teams should be doing the central analyses in parallel. And then there may be secondary analyses that follow kind of the primary discovery effort in an attempt to annotate function using bioinformatic analyses, or even performing functional follow-up experiments that might use model systems, either cellular or you know, potentially even organismal if you have high throughput whole organism assays like zebrafish. And so these might be their own working groups since they have different expertises and functions. And then lastly, a writing group, a group who's tasked with the job of summarizing the results, both writing up the methods, collating a fair amount of cohort level data, and then organizing them in a manuscript that can ultimately be circulated to all the members of the consortium. But the writing group, you know, a typical functional writing group might include, you know, 5 to 6, maybe 8 people, commonly comprising the analyst and PI from each of the 2 central analytic groups, but potentially others who may have a pretty prominent role in some of the secondary analyses, for example.
Chris, can you talk later about how kind of how the format of your data resources affects how these groups work? Like, I can imagine that cloud computing has really, and cloud storage has really changed some of your organization for these large studies.
Yeah, so we will. I will talk a little bit about that, but I think you're right that how like the physical location of the data and the deposit process, obviously that has changed over time. So I'll come to that. I'll touch on that in a bit. I'm just gonna make a little note to myself.
Thank you.
You know, when thinking about why have 2 central analytic sites that are performing identical work for, say, the primary association analyses and QC of the data. Well, a con is clearly it's double the work. It's 2 groups having to do exactly the same thing. And so to some extent, reinventing the wheel has a redundancy to it. And it can slow the forward progress of the project due to the need to coordinate between 2 sites. But advantages of having 2 sites include the ability, it enhances quality control, because essentially you have the ability to compare both quality control evaluation and association results. And when there are discrepancies between the two sites, then in the process of resolving those discrepancies, it typically leads to recognition of, you know, either a bug in the software that might be used, or a formatting problem in the data. And commonly there's a fair amount of reformatting that may be required depending on the workflow that's been established. So in fact, the redundancy gives some protection, and, you know, clearly it can be the case that, you know, primary analyst may become ill or have a life event, you know, marriage, pregnancy, might leave the institution, might have family issues arise. So there is a threat to the overall project of consolidating too centrally the analyses. It also promotes transparency, because rather than having a single analyst who's producing QC and results on their own, where they might, you know, take shortcuts of things that are familiar to them, this actually requires that both analytic sites kind of explicitly describe what they're doing. And this explicit record keeping and documentation can be very helpful down the line. Some of these projects will take can take years, and, you know, in the revision process, often going back to determine what exactly was done at different stages, I think is promoted by having the two sites. And finally, you know, as anybody who, you know, let's say you're a morning runner, there's a difference between setting your alarm and getting up and going for a solo run, and setting an alarm and meeting another person who is going to run with you. And I think that kind of helps promote staying on time and maintaining forward momentum of the project, which you know, because everybody obviously can have a whole set of priorities that can overtake their contribution to a project. You know, I think it's really important at the outset of a project to be quite formal in developing a detailed written analysis plan. And you know, I think often these can be modeled on other projects that have had a similar structure, but a detailed listing of the quality control steps that should be applied at the cohort level. For example, things like imputation backbones that should be used, whether to use 1,000 genomes or you know, various iterations of UK Biobank or TopMed or other sequencing projects. What analytic steps need to be undertaken, including things like inclusions and exclusions, and filtering of the individuals and the phenotype data. And then specifically what software tools and versions of those tools are recommended to be used. And you know, it's definitely not uncommon that two sites might be using different versions of the same tool and have slight differences that result as a consequence, most commonly in formatting. One needs to specify the format of the results return, and this can really reduce the burden on the central analytic team when they're receiving data, but it is an opportunity for mistakes to arise if the formatting is not handled correctly. And then even just things, you may have analysts involved at a given cohort who actually are very unfamiliar with the phenotype and unfamiliar with the types of quality control or recognition of impossible values or other issues. I've done studies where it was a quantitative trait, but it was coded as or it was a continuous trait, but it was coded as a 0-1 trait and not recognized. You know, these things, obviously they should break your software machinery, but it's quite important. Or you know, they're on the standard deviation scale versus on the natural raw measured scale. So things like collecting phenotype medians and standard deviations and covariate effect estimates can give, you know, if one knows that, say, you know, diabetes is associated with higher blood pressure, and the effect estimate for diabetes shows protection against higher blood pressure, then that might suggest that there's some issue with the coding. You know, I think the classic would be faulty coding of case and control status for a case control design, where essentially you get all results that are in the opposite direction that is the reality for all the other studies that have properly coded them. And lastly, I think it's important to specify an analysis plan in part because one wants to collect as much of the data at the outset that will be needed to finish the project if possible, because I think, you know, for a project that may take a couple of years, it's possible that the people will have moved on, that the personnel involved in the project will have changed at a given cohort, and so going back iteratively runs the risk that that different people are involved and are pulling data from different places. So data storage and analyses, you know, centralization of data deposit, typically one location to deposit data, and there are, you know, cloud solutions now that facilitate this. In the studies that I'm directly supervising, I have not used cloud deposits. So I can't specifically speak to that aspect of it, but the analyses can certainly be performed in the cloud, and I think performing these analyses in parallel is really important, and so that means that two different people really should be coding and recoding the data independently, rather than, you know, if one person just sends their the same code that they use to run an analysis, obviously that should be done after the fact, but you don't want, you could make the same mistake twice if both groups are doing, I should say, if they're not independently setting up their pipelines for performing the analyses. And there's some streamlining of this, and there are packages that obviously handle a lot of the start to finish. But clearly there are multiple stages of these projects and multiple iterations where there are outputs of earlier analyses. And so I think, you know, one common deposit site where all of the cohorts deposit their data, and that represents a source of truth for the original submitted data that is not modified by the central analytic group, and then parallel file storage and reformatting and analysis of files as necessary done at the two sites, with and then that's where the analyses are performed, and then there is typically a separate site for location of results summaries, manuscript files, presentations, meeting minutes, other very kind of low data size, low processing need, like Dropbox or Google Drive or other mechanisms for exchange of summary results and presentations. So it's important to set timelines, and I think commonly we'll set dates for freezes of data deposit, rather than waiting until all data has been submitted and then starting analyses at that time or starting QC. It's much better to do them iteratively, so that if problems are systematically arising, one can reach out to the entire group and make sure that there's clarification of where the problems are, or any modification of the analysis plan. You have to recognize, obviously, there can be slippage in these timelines, but that slippage is really a threat to the overall success of the project. And so I think regular steering committee meetings where the working groups report up to the steering committee about status of their individual work efforts, something on the order of once a month, depending on how active a group is. And then there needs to be some form of periodic all cohort meetings. And it's important to minimize long gaps in update. I mean, it can be, some of these can be teleconferences or, you know, Zooms, and some of them can just be email updates, but it is important as part of one's kind of consortium hygiene to make sure that the people who have been contributing work even if they're not actively involved in the central analysis, know that there's been forward progress. And I think the central analysis group needs to have regular meetings, probably on the order of one time a week, in addition to the primary analyst and supervisor meetings that will be happening at each of the sites. And I think that A, that redundancy, and B, the frequent meetings to compare notes on what's going on are ways in which problems in the data can be identified. And then lastly, writing group meetings will occur and require very explicit delegation of who is working on which aspect. These projects typically are quite large and have many working parts, and it's important to parallelize work when appropriate, rather than stacking everything on the backs of, for example, the primary central analytic analysts. So this is just, I'm sure this seems very stupid and maybe how to run a successful meeting. Certainly my kids going through high school have had a fair amount of training in how to give a presentation and how to stay on point and have a meeting. But I think it's useful to kind of use a business world framing. And the way I view any meeting is, you have X number of people in the meeting. Maybe there are 10 people on the call. If you have a 5-minute waste of time, then that translates to 50 person minutes that have been wasted. If you have 100 people on a call, if it's a large group forum and you waste 5 minutes, you've wasted 500 person minutes. And it's really, you need to respect the time of the participants in the meeting, have meetings that are ended early when the meeting goals are accomplished. Don't just fill the time. One of the ways to avoid that is having a very detailed agenda in advance of the meeting, which has the advantage that participants will show up prepared and will know it's being discussed, or depending on the subject or data being discussed, will have a representative from their cohort who's more familiar with those be present. It's important to articulate in the agenda who's going to be presenting what in advance. Ideally, designate in advance who's chairing the call and who's taking minutes. It's useful for those to be two separate people, especially for larger meetings. It can be helpful to allocate approximately the amount of time that will be spent on the different components. I've definitely been on poorly managed consortium calls where there might be 4 agenda items and the first item takes 45 minutes and the last 3 items get packed into the final 15 minutes, and someone who came prepared with a presentation that would have taken 10 or 15 minutes is then forced to shrink it to 1 or 2 minutes or update the group offline. So respect the participants and presenters by giving a certain amount of time, and if you reach the end of that time and need to move on, then you move on. And I think it's important to do that. That's where a chair of the call can be very helpful. I've already said, value the time of the participants, both the presenters and the recipients of the information. And circulating minutes after the call can be very helpful for people who are unable to make the call, and also represents a point in time reference that people can use to understand the stage of the project. Writing groups. I can't highlight enough the importance of delegating explicitly. Groups of people don't get things done in a collective sense. Individual people who are members of a group get things done, and there's nothing more frustrating than 2 people doing the same thing when only one person is required, or no person working on something because each thought the other was working on it. It is never too early to write up methods. So I think in the same way that an analysis plan is a forward-looking document that that describes what is intended to be performed, it is helpful for the people performing the analyses to periodically summarize their methods. Sometimes it's not apparent exactly what is being done to the group, and this will sometimes force clarification of issues rather than waiting until the project is largely complete and then writing up the manuscript and realizing that there's some systematic issue along the way. Typically, the writing group will comprise at least the lead central analysts and their supervisors, but often others who may have more prominent secondary analytic roles or responsibilities. Something on the order of 5 to 10 people is a reasonable number, but obviously the primary writing is typically done by the lead analysts for at least initial drafts. So with all this great planning, what could possibly go wrong? Well, I'm going to talk a little bit about some of the common pitfalls that arise. Well, number one is timeline slippage. Really important to acknowledge holiday breaks. The amount of work that gets done between December 15th and January 15th is really limited. Summer breaks are not aligned at many countries. You know, maybe the habit is everybody in France goes to the beach in July, and everybody in Finland, you know, goes to Lapland in August. It can be very challenging, but the summer is a particularly slow period for very distributed work. So if you're waiting for data sets to be deposited and your freeze follows the summer, expect to have a lot of rolling submissions as opposed to the rest of the year. And then things like illness will arise. People are, you know, defending theses, marriage, pregnancy, pandemics, like things come up, and it's important to recognize that those things will happen, and, you know, have a good plan for how to keep things moving forward and identify when things can't move forward. Say if the primary analyst, usually you know when you're getting married, so you know when you're going to be needing time afterward or planning before. But many of these things can come up by surprise. Additional cohort level analyses are really challenging. Sometimes one you know, is writing up the paper and there's a secondary analysis that's felt to be desirable, you know, if you have to go back to all of the cohorts which may have submitted their primary data, you know, a year and a half earlier, and now you're asking them every cohort to do another analysis, well, there's a chance that it's going to be different people at some of those institutions. And especially as the cohort numbers increases, let's say 10 cohorts versus 50 cohorts, so does the amount of time for the last cohort to submit any new data that is requested. And the larger the project, the greater the threat if complete data submission is required. One alternative is to consider performing secondary analyses just in a subset, for example, in a relatively smaller number of larger cohorts that might be sufficient to ask some of the secondary questions. That reduces the analyst burden at the cohort level. Collecting authorship information can take forever, and you know, you can collect it at the outset, but unfortunately, at many cohorts or for many of the secondary analyses, you won't know at the start of the project exactly who's going to be involved. Their names change, they get degrees along the way, their affiliations may change, their email address may change, they may have funding acknowledgements that are required if they have a training grant, for example, that pays for their contribution to the project. And chasing down delinquents can often have to be pretty creative. And I know that in the summer, I remember needing, you know, Nature Genetics forced us to get, you know, explicit permission to add co-authors to the paper from every one of the 300 co-authors. And I remember having to call a friend in Finland to ask them if they could text or in some way communicate with someone to get explicit permission. You have to be pretty, not everybody is on email all the time. And so it's good to have ways to track people down. But the authorship part, it always takes longer than you think. And so usually by the time you're assembling a writing group, that's a good time to be soliciting authorship information. Authorship conflicts can come up, and these can be pretty stressful. You know, I think it's not uncommon for large consortial papers to have stars, but deciding which of the first or which of the last authors have contributions that warrant stars can be controversial. The author order at the beginning or end or throughout the manuscript can be controversial. Who at some institutions or in some countries, who the corresponding author is matters, and others it's irrelevant. So you have to recognize that there are different incentives and priorities in different academic environments that can vary by institution or by country. One needs to balance contributions across institutions, especially when the workload is shared. Although if it's disproportionate at one institution, then maybe when that occurs, unless it's required because of you know, certain experiments or something that's being done there, it's better to try to balance the work across institutions. I would say resolving these conflicts can be challenging. It's best mediated by supervisors and by the steering committee. And so it's helpful when the steering committee is not only people who are directly supervising all of the conduct of the work, because then there aren't so many kind of third-party members who can comment on exactly who contributed what or reflect that. And I think these conflicts can be reduced if explicit criteria or principles of authorship are established up front that can serve as a guide. And then there can be a breach of transparency rules. Consortium members, you know, do work together and they do work apart. So use of aggregate data in independent manuscripts really should be spelled out explicitly, and notification of the cohort of the consortium of work that's happening is important. Use of individual cohort data by a central team for a separate project is a definite no-no. So it may be that a cohort contributes data for a specific analysis. That doesn't mean that necessarily, depending on the terms of the consortium, that they're agreeing that that could be used for other projects that are not explicitly approved. And you know, I've experienced one where a cohort might contribute to multiple consortia, including for the same analyses, potentially leading to competing manuscripts where the same data are being presented in more than one simultaneous submitted manuscripts. And as a last resort, it's, you know, if someone has an infraction of the terms of participation, there can be expulsion from a consortium. Having participated in some of those discussions and observed that, I could say it's really messy and stressful, and you really are glad you have a steering committee that has had regular input and updates about what's going on in the project at those times. Departure of a central analyst. You know, projects can take years. Personnel may change their institution or their career path. Now, I've had one person who was in academic environment and then went to industry right at the middle of the project, and it was pretty stressful. Dual analytic sites really provides important redundancy. You know, if the supervisor remains, they can often identify another trainee or identify potentially a new site to carry out analyses. It's important to define an exit process. Maintenance of an electronic lab notebook, which should be, you know, standard, I think can be quite helpful for helping a new analyst on board to take over, and really documentation throughout the course of the project about what is being done is really good insurance in case a central analyst has to leave the project. Inadequate communication certainly can arise and lead to frustration. You know, from the time cohorts submit data to preparation of a manuscript can be relatively long. Failure to keep the consortium apprised of progress can be irritating to members. The steering committee can suggest cohort communication updates, you know, and kind of remind the people who are down in the trenches doing the work, you know, maybe this is a good time to give an update to the consortium. Presentation at both consortium meetings or presentation of abstracts at scientific conferences can be important ways to guard against the communication problem and keeping everybody in the loop, and also generating enthusiasm for the progress of the project. There can be trainee supervisor conflict, and this is challenging. The PI, you know, often has grant funding, has a secure faculty position and multiple publications with prominent authorship, whereas the trainee has finite effort, has a need for publication and funding, and has uncertainty about their future. So they have really different incentives. And, you know, there can only be one first author and one last author, even if all the stars are supposed to distribute that effort, and different institutions consider stars variably around the world. So it's important to have open discussion of the trainee and the supervisor, and it's really important for trainees to have external mentors whom they can use for advice. I will say there are a bunch of benefits to the trainee. Number one, you gain experience presenting to large groups. You have involvement in high impact projects. It facilitates networking, which can include future offers of postdoc or faculty positions. That's probably the greatest benefit, is it really exposes you to a large universe of people in your field, and it can help you develop project management skills. But you really need to balance your consortium work with small group focus projects where your contribution is a much greater proportion of the overall work and reflected in authorship accordingly. Eventually, you get to a large body of work or some recognition or grants, and hopefully this discussion of some of the process is helpful. I'm happy to answer questions that folks might have, and maybe take it off screen share, and I guess, is there a gallery view?
Thank you, Chris. That was a great talk. And I realized we forgot to acknowledge that for years you ran this primer session.
Oh,
so
yeah.
To have you back.
Yeah. So any questions or comments?
One thing I was curious about was when you were discussing return of results. I know that kind of standards for returning secondary findings have changed greatly, probably throughout the time that you've been running such studies. And how has your group decided when to retroactively change secondary study reporting, or have you always just stuck with the initial intent of the study?
This is a big long-term issue for these types of studies, especially when you have consortia that may have initially had a bunch of participants who had a certain consent for their contribution of data. The reconsenting process is quite laborious, and in fact there are different participant protections by institution, by country, and different standards. So it can be challenging. I would say in general there's a balance between the understanding of the participants at the time that they consented to the study and the urgency of informing people who may be at risk. And there have been various strategies to deal with those kinds of secondary or subsequent discoveries that might be quite impactful for individual participants. There's a question from Jose. Curious to hear your approach on the timing at which authorship is decided, negotiated up front, or after you see what everyone has really contributed. So I think the declaration of the authorship principles is really optimally done up front. And, I think that is different than identifying the individual people who will could contribute to a project. And so one of the principles should be clearly articulated at the outset, which is authorship prominence in proportion to contribution to the overall project. That's just, you run the risk if you designate first and last at the outset of the project, that you demotivate other people to contribute as much to the project. But the alternative is if you don't indicate who is going to have a leading role at the outset, then it can be demotivating to work hard. So I think there's a balance, and I think different consortia have handled these things differently. I'm definitely aware of some consortia that have identified who the first, the two first authors and two last authors will be at the very outset. But I've been in projects where the authorship criteria are pretty well established. The consortium has a track record of mediating authorship disputes, and there's a general comfort that the work performed will be honored in the assembly of the authorship sequence. But I've definitely had quite contentious authorship discussions that have delayed submission of manuscripts, and I think we've all heard various horror stories.